Hello ISMB, my name is Leal. I am a research scientist in Novo Nordisk Research Center, Oxford. Together with Joanna Sharman and other colleagues at Novo, we developed gene prioritization algorithms for target discovery in complex diseases assisted by machine learning-driven data weighting. Gene prioritization or gene ranking is the process of algorithmically ranking genes based on their predicted involvement in a trait of interest. The methods typically integrate layers of biological evidence to generate an overall ranking. Such ranking can help you reduce long lists of targets and highlight promising candidates for validation. Despite of the vast literature on this topic, there remain challenges to the process. A first challenge is to understand the weighting of biological layers, how to select a suitable weighting with or without expert field bias. A second challenge is to ensure that prioritized targets have a balance between disease evidence and novelty. Here, novelty refers to lack of prior literature for a gene in the disease of interest. We still don't have methodologies to guide the construction of novelty scores and allow comparison with rankings from biological layers. A third challenge is to tailor rankings towards traits of interest that can be experimentally tested. Gene disease scores can be too general, and the genes that are important to specific traits need to be identified so that they can be tested in relevant experimental assays. Given that not all data sources are relevant to the biology tested by the experiment, we require a feedback approach from the experimental part to improve our algorithms. Here we present two algorithms for integrating gene disease evidence and describe the application to drug target discovery to address these three challenges. The algorithm has been evaluated in the cardiometabolic disease space, and we present a use case in atherosclerosis. We collected 7 different layers of biological evidence for association in atherosclerosis, including gene expression, proteomics, literature data, in vitro data, genetics information, knowledge graph-based features, and pathways. We developed 2 algorithms. The first algorithm, here referred to as BASIC, is designed to score each biological layer based on user-defined weights. To avoid bias, we typically set each data source to contribute equally to the overall score. Internally, the basic algorithm uses data transformation functions specific to each data type. The raw data is transformed into the same interval of values between 0 and 1, and this is later aggregated to score every gene independently. The second algorithm, KCCA, kernel canonical correlation analysis, follows previous work for transforming genomic data into gene-gene similarities and correlate those similarities within a low-dimensional space, canonical space. This technique learns the weighting of the biological layers or the feature importance by maximizing the correlation of benchmark gene-gene similarities across data sources. Our benchmark gene set for atherosclerosis includes 304 targets reaching preclinical or clinical pharmaceutical development. Higher ranking is given to targets closer to the benchmark in the canonical space. These violin plots present a gene list versus a background of 21,000 genes. The scores should be interpreted together with the data completeness score. The more data, the more confidence in the ranking. Also, the probability of a random score. The lower the probability, the less likely the ranking changes. We are making the basic algorithm available in our external tool called Target Credibility Indicator, TCI. This is the demo of our external tool. It is used for prioritizing targets for layers of atherosclerosis, and the release includes 4 layers: in vitro layer, genetic data layer, protein expression, and pathways. The user can select their own weights and upload their own gene list. After clicking on Compute Scores, we query pre-computed data according to the weighting, and the table below can be used to select the different metrics for visualizing the rankings: the gene score, the data completeness, and the probability of random score. There is also an option to download the scores to a TXT file. In the Visualization tab, the user can compare the gene list versus the background in the overall score and in each of the layers. There is also an option to compare a single gene versus the background. In this case, we compare the LDL receptor in the in vitro layer. The density plots will show the position of the gene versus the background of 21,000 genes. In addition to the scoring of genes per biological layer, we incorporated a novelty tiering system from the least novel level 1 to the most novel level 4. In level 1, we have existing drug targets in clinical development for the disease of interest. Level 2, when the gene is under internal evaluation. Level 3, whether there is significant literature evidence linking the gene to the disease, and level 4, when there is no significant literature evidence linking the gene to the disease. This tiering enables prioritization of novel candidates that do not reach high overall scores but still rank top in human-centric layers such as in vitro disease models. In this example, a gene list is ranked with the layers contributing as shown in these pie plots. The basic algorithm gives an equal weight to all the layers by the use of defined weights, whereas the KCCI algorithm learns the weights using machine learning based on the benchmark gene set. For this gene list, particularly the pathway layer contributes the most. The ranking was compared to the results from testing the genes in vitro assays. The box plots present a higher ranking for the hits in green versus the non-hits in gray, but these differences depend on the biological layer, the assay readout, and also the algorithm. In summary, we approach the gene prioritization by interpreting biological evidence in two ways. With the basic algorithm, the evidence is additive and linear. The weights are user-defined and the genes are scored independently. With the KCCA algorithm, the evidence is aggregated by way of gene-gene similarities, the weighting is machine learning driven, and the ranking is specific to the input gene list and the benchmarks. Second, the novelty tiering system adds extra functionality for balancing novelty with gene disease evidence for drug target discovery. And third, we demonstrated that atherosclerosis rankings are concordant with the assay results and specific layers are more highly correlated with the hits. Thank you very much for listening, and I hope to see you again on Monday, July the 24th during the first poster session.